Assalamu alaikum students. Today we will discuss about the organization of the eukaryotic chromosomes. Eukaryotic genome are the more complex as compared to the prokaryotes. Mostly the eukaryotes exist in the diploid form and there are some problem plants which are the polyploid. Although eukaryotes have only about 2 to 15 times as many as genes as E. coli, they have an order of magnitudes more DNA. Moreover, much of this DNA does not contain the genes because more if we describe the genes must be a functional unit which may be involved in the production of proteins. DNA is also packaged in the eukaryotes. DNA is also packaged into the several chromosomes. So in the eukaryotes, the genes are also present in in the compact form. So the chromosomes in the eukaryote is actually in the compact form. Eukaryotic chromosomes contain 1 to 20 centimeters of the DNA, and during the process of the divisions, like the meiosis and the mitosis, it reduces up to the 1 to 2 micrometers. These chromosomes contain single giant molecule of the DNA, which extend from end to the centromere and to the centromere to the other end. And this is the highly condensed molecule. Okay. There are the three levels of the DNA packaging in the eukaryotic chromosomes. This is the electron micrograph of the nucleosomes. Uh, Largest human chromosomes contain 85 millimeters, or you can say 85,000 micrometer or 8.5 uh, nanometer of the DNA. In metaphase structure, this chromosome is 0.5 micrometer in diameter and 10 micrometer in the length. Under the electron microscope, the DNA chromosome is found as ellipsoidal beads joined by the thin thread. Further evidence for the DNA are studied by using the nucleases. Partial digestion yield fragments of the DNA of various sizes. These beads are called the nucleosomes according to the present concept of the chromatic structure. The linker or the interbeads uh, thread of the DNA are susceptible to the nuclease attack. Okay, if you can see in the beads, just a minute. If you can see in the beads, beads are actually wounded in the beads. DNA is actually wounded on the histone protein, and in the linker DNA, mostly the DNA is naked. So that's why this region is more susceptible to the nucleases. Eukaryotic chromosomes contain a long linear DNA molecule. Three types of the DNA sequences are required for the chromosomal replication and disaggregation. Number one is the origin of replication, from where the replication uh, is started. The second one is the centromere, which is actually the central part of the or the mid part of the chromosome, and third one are the edges of the chromosome, which are called the telomeres. This is the chromosomes. Here you can see these are this edge. These are the edges, which are known as the telomeres. And this is the center, which is the centromere, and this is the origin of replication. And in between these origin of replication and the telomere, genes are spread. Eukaryotic chromosomes are usually linear. A typical chromosome is tens of millions of hundreds of millions of base pairs in the length. They are very large molecules. Each chromosome contains many origins of replication. Each chromosome contains centromere and it contains the telomeres on the edges. There are repetitive sequences which are commonly found near the centromere and the telomeric regions, but they may also be intercepted throughout the chromosome. Genes are located between the centromeric and telomeric regions along the entire chromosomes. In lower eukaryotes, such as these genes are relatively small. They contain primarily the sequences encoding the polypeptides. Very few introns are present in the lower eukaryotes, but in the higher eukaryotes, mostly the genes are long and they tend to have many introns, and they have many in intron regions spread in the chromosome. Repetitive sequence complexity refers to the number of times of a particular base pair sequence appears in the genome. There are three main types of the repetitive sequences: unique or the non-repetitive, moderate repetitive, and highly repetitive. Unique repetitive, which are found once or a few times in the genome. They include the structural genes as well as intergenic areas. Moderate repetitive found a few hundred to few thousand times within the genome. They include genes for the ribosomal RNA, stone proteins, or origin of replication, transposable elements. Now, the last one is the highly repetitive sequences. They are tens of thousands, millions of times repeat in the genome. Each copy is relatively short, and some sequences are interspersed throughout the genome. Example, allele family in the human. Other sequences are clustered together in tandem arrays. So, for example, AATAT and AATATAT sequences in the Drosophila. These are commonly found in the centromeric region of the chromosome. After denaturation, we know that DNA stands at high temperature at denatured, and after getting a lower temperature, they are again renatured. Denaturation experiment. The rate of denaturation of the complementary DNA strands provide a way to distinguish the three different types of the repetitive sequences. The denaturation rate of the particular DNA sequence depends on the concentration of its complementary part. Uh, repetition experiments provide the evidence that uh, highly repetitive sequences will rebind with each other at the higher rate, moderate at the higher rate, or you can say highly repetitive DNA will be the fastest to renature because there are many copies of the complementary sequences. And there is very low rate or very slow procedure for the renaturation for the unique sequences. This is the experiment. Here you can see the renaturation of highly repetitive sequences is very high, and for the moderate sequences, it is lying somewhere in the middle, and the unique sequences are very slow to denature. And 60 to 70% of the DNA, human DNA, are unique sequences. Eukaryotic chromosome compaction. Uh, as for we discussed in case of bacteria, that the chromosome is larger than the or we can say that the chromosome has a more length as compared to the space in the cell, so it need to be compact, it need to be shortened down, it need to be compact. Like that chromosome, human chromosomes also need the compaction. If stretches end to end, a single set of the human chromosome will be over 1 meter long, yet the cell nucleus is only 2 to 4 micrometer in the diameter. Therefore, the DNA must be tightly compact to fill. The compaction of linear DNA in the eukaryotic chromosome involve interaction between the DNA and the various proteins. These proteins bind to the DNA and are subject to change during the life of the cell. Nucleosome. Nucleosome is actually the beaded structure. It is actually the interaction between the DNA and the proteins, the repeating structural unit within the eukaryotic chromatin. This is known as the nucleosome. This is composed of double-stranded DNA wrapped around the octamer of the protein. The protein is known as the histone. An octamer is composed two copies, each of four different histones, and 146 base pair of the DNA make 1.65 negative superhelical turn around the octamer. This is the histone protein. If you can see, nucleosome uh, contains the 8 histone proteins plus 146 nucleotides base pairs of the DNA, which are wounded on these histone proteins. The diameter of the nucleosome is around the 11 nanometers. Um, to link the two to link the two units containing the histone proteins and the DNA, there is a linker DNA which make the link between the two beads. It is composed of the 20 to 100 base pairs of the DNA and this length varies species to species, or we can say this is depending upon the species. Histones play a major role in the chromatin. They are present in the chromatin of all the eukaryotes in amount equivalent to the amount of the DNA. This relationship suggests that an interaction occurs between the histones and the DNA that is conserved in the eukaryotes. The histone of all plants and animals consists of five classes of the proteins: H1, 2A, 2B, H3, and H4. So there are two H2 units which are further subdivided into A and B. Four of the five types of the histones are specifically complex with the DNA to produce the basic structural subunit of the chromatin. Small ellipsoidal beads. These beads are called, I have already told you that these beads are called the nucleosomes. The histones have been highly conserved during the evolution. Four of the five types of the histones are similar in all eukaryotes. Most of the 20 amino acids in the proteins are neutral in charge. That is, they have no charge at the pH. However, few are basic and few are acidic. The stones are actually the basic because they contain 20 to 30% arginine content and lysine. Two positively charged amino acids. So because of these two positively charged amino acids, because arginine and lysine are making the 20 to 30% of the histone protein, so mostly the charge in the protein is so the histone protein is actually the basic. Exposed amino group of the arginine and lysine allow histone to act as polycations. Exposed amino groups. These positively charged side groups on the histone are important in their interaction with the DNA, which is polyanionic because of the negatively charged phosphate groups. Remarkable consistency of the histones H2A, 2B, 3, and 4 in all cell types of an organisms and even among wildly divergent species is consistent with the idea that they have or they are important in chromatin structure and are only non-specifically involved in the regulation of the gene expression. So histone protein is not only involved in compaction of the DNA, this is also involved this is also involved in the regulation of the gene expression. There are some chemical modifications on the histone protein which can alter the chromosome structure, which in turn can enhance or decrease the level of expression of the genes. Okay, you can say by change in the histone structure, the compaction or relaxation of the chromosomes takes place, which will allow the expression of genes or which will allow to stop the gene expression. Moreover, the composition of the non-histonic chromosomal protein fraction varies widely among different cell types of the same organism. Thus, non-histone chromosomal proteins probably do not play a central role in the packaging of the DNA into the chromosome. So because other protein which are non-histonic proteins, they are also playing role in the compaction of the chromosome, but their fractions vary widely among the different cell types. So they have some role in the compaction of the chromosome in the packaging of the DNA, but central low role is definitely for the histone because it's not varying; it's constant in all types of the cells. This is the structure of arginine and lysine. You can see that they are containing the positive charge. Lysine because of the extra amide group, and arginine again for the extra amide group. They are containing the positive charge. Histone protein being basic, they contain many positively charged amino acids. I told you that lysine and the arginine are the basic amino acids which are present in them. Uh, H1 is the linker histone protein. H2A, 2B3, and the 4. These four proteins are the core histone proteins. Each contain two units for interacting with the DNA. So these four protein 2A, 2B, H3, and H4 uh, are present in their two units, are present and having their two units. So making the octamer. Remaining the fifth protein H1 is actually the linker histone protein. It binds to the DNA, also bind to the nucleosome, but not as tightly as the core histone. As you can see, and this is the diagram. This the central part is the uh, octamer, and 146 base pairs are rounded on this octamer protein. Here you can see the H1 histone protein is clipping the DNA bounded on the histone protein. You can see. So for example, if this is the histone protein and here the DNA is bounded on the histone protein, say for example, this one is the 4 in two forms. So this is, but this one is the octamer. This one is the octamer, and here the linker histone protein is present to clip the DNA or to pack the DNA tightly with the chrome with the histone. Okay, chromatin and nucleosome. These are the nucleosome. These are 11 nanometer in length. After after the nucleosome, or you can say after the interaction with the nucleosome, chromatin fiber stretches during the preparation for the electron microscopy, revealing linker DNA between the nucleosome core. Linker DNA vary in length from the, say for example, 8 to 114 or 20 to 100 nucleotide base pairs. Nucleosome core 146 nucleotide pairs of the DNA wrapped as one uh, into 3-4 turns around an octamer of the histone. If you look on this diagram, say for example, this is the DNA. This is the compact structure of the DNA wounded on the histone proteins in the chromosome. If we stretch this structure, we will obtain this type of the electron micrograph. Here you can see this one is the linker DNA, and and the beads these are the beads containing the bonded DNA. So the linker DNA, I told you that its length must be 8 or 20 to 100 or 140 million. In the nucleosome, one complete turn and 1 into 3 over 4 complete turns are in 1 into 3 by 4 turns containing 146 nucleotides in the DNA are wrapped on the histone protein. These nucleosomes further interact with each other to form a 30 nanometer fiber to make the DNA more compact. Nucleosomes associated with each other to form a more compact structure termed the 30 nanometer fiber. This compact structure is known as the 30 nanometer fiber. Histone 1 protein play a role in this procedure. A moderate salt concentration when the H1 is removed, the result is the classic bead on a string morphology. So when we increase, when we further decrease low salt concentration, H1 remains bound. Beads associate together into more compact morphology. So if we moderately treat, if we treat the histone 1 with the moderate salt concentration, they will removed and the chromosome cannot get the compact structure. But at low salt concentration, they remain bounded form. They remain in the bounded form, and if they remain in the bonded form, chromosome will be more compact. 30 nanometer fiber shortens the total length of the DNA another 7 folds. Its structure has proven difficult to determine. DNA conformation may be substantially altered when extracted from the living cells. Two models have been proposed, solenoid model and three-dimensional zigzag model. The structure of the DNA conformation may be substantially altered when extracted from the cells. During the extraction, the structure is altered. Two models have been proposed. One is the solenoid model and second one is the three-dimensional zigzag model. These two models are made during the formation. These two models are very important to explain the chromosomal structure. This is the solenoid model for the 30 nanometer chromosome. These are regular spiral conformations containing six nucleotides per six nucleosomes per turn. The three-dimensional model, this is the zigzag model. This is also the 30 nanometer fiber. This is actually the irregular configuration where nucleosome have little face-to-face in phase contact. The two events we have discussed so far have shortened the DNA about 50 folds. A third level compaction involved interaction between the 30 nanometer fiber and the nuclear matrix. The nuclear matrix is composed of two parts, nuclear lamina and internal matrix protein. 10 nanometer fiber and associated proteins also involved in this compaction. Okay. After making this, the cellular protein will interact with the DNA with the chromosomes to make it more compact. Here you can see the matrix. You can see the matrix protein bounded to the 10 nanometer fiber. This is the 10 nanometer fiber. These are the matrix proteins. This is the nuclear lamina. This is outer nuclear membrane, inner nuclear membrane, and the nuclear pore. So you can see that matrix proteins are bounded to the 10 nanometer fiber, and this is the binding of the proteins with the fiber. These are also important compaction of the DNA structure. The third mechanism of the DNA compaction involves the formation of radial loop domain. And this type of domain is formed after the interaction with the uh, matrix assisted proteins. Matrix proteins. The loops are formed. Here you can see these loops are known as the radial loops. These loops contain 25,000 to 200,000 base pairs in base pair. These are actually the matrix assisted regions which can interact with the matrix protein. MAR's matrix assisted proteins are anchored to the nuclear matrix, thus creating radial loops. Further compaction of the chromosome, the attachment of the radial loops to the nuclear matrix is important in two ways. It plays a role in the gene regulation. It serves to organize the chromosome within the nucleus. Each chromosome in the nucleus is located in a discrete and non-overlapping chromosome territory. Here you can see the compaction level of the interface chromosome is not completely uniform. There are two compaction levels. One compaction is the euchromatin. These are the less condensed region of the chromosomes. Transcriptionally active chromosomes in transcriptionally active regions. It means the mRNA can be made from here when the chromosome is in its eukaryotic, it's in euchromatic form. Regions where 30 nanometer fiber forms radial loop domains. Heterochromatin, these are the tightly compact regions of the chromosome. Transcriptionally inactive genes cannot function and radial loop domains compact even further. This is the compact diagram of the chromosome. You can see this is the complete chromosome with the three regions: origin of replication, telomere, grandiose, and telomere. There are two types of the heterochromatin. There are two types of actually the heterochromatin. One are the constitutive heterochromatids, regions that are always the heterochromatic and permanently inactive with regards to the transcription. And second one is the facultative heterochromatin regions that can interconvert between the euchromatic and the heterochromatic, like the bodies. This is DNA double helix. This now the regarding the summary of all the compaction procedure. This is the DNA double helix bounded on the nucleosome. These nucleosomes are further uh, you can say that DNA when bounded with the histone protein make the nucleosomes. These nucleosomes will interact with one another either in the solenoid model or in the zigzag model. In the solenoid model, the DNA are face to face and close to each other, and while in the zigzag model, DNAs are irregular, not regular. Nucleosomes are irregular, not regularly arranged, and they are not facing each other. After making the zigzag structure, radial loops are formed. Loop domains are formed by the interaction with the uh, matrix-assisted proteins. Radial loops are formed. Further, these radial loops are compacted into the heterochromatic or the further they are compacted into the heterochromatic form during the interphase. Most chromosomal regions are the euchromatic, but for its further compaction, the chromosomes are heterochromatic, and they are interacted with the matrix-assisted proteins, and then finally in the metaphase, the chromosome is fully compact structure, um, and make after making the scaffold folds from the nuclear matrix and the further compaction of the R radial loops. Metaphase chromosomes as a cell enter and phase, the level of compaction changes dramatically. By the end of the prophase, sister chromatids are entirely heterochromatic. Two parallel chromatids have an over a diameter of the 1400 nanometer. These highly condensed metaphase chromosomes undergo little gene transcription. In metaphase chromosome, the radial loops are highly compacted and stay anchored to a scaffold. The scaffold is formed from the nuclear matrix. Histones are needed for the compaction of the radial loop. Two multi-protein complexes help to form and organize metaphase chromosomes. One is the condensin, which play a critical role in the chromosome condensation. The second one is cohesin, play a critical role in sister chromatid alignments. Both contain category of proteins called SMC proteins, which means structural maintenance of chromosome. SMC, structural maintenance of chromosome, SMC proteins. SMC proteins use energy from the ATP and catalyze energy in the chromosome structure. Here you can see the condensation of the metaphase chromosome by the condensin. During interphase, condensation is in the cytoplasm. This is 300 nanometer radial loop chromatin, radial loop or the euchromatin. When this is because before the G1S and G2 phases, start of the M phase, condensins travel into the nucleosomes. Here you can see during the interphase, condensins are in the cytoplasm, but during the M phase, condensins move to the nucleus and bind with the chromosome. 700 nanometer heterochromatin and condensins. You can see condensins are bounded with the chromosomes. Condensins bind to the chromosomes and compact the radial loops. The number of loops has not changed. However, the diameter of each loop is smaller. Alignment of the sister chromosomes through the cohesion, end of the S phase and G2, uncondensed sister chromatids arms are cohered. Uh, beginning of the prophase, during the beginning of the prophase, condensed sister chromatids arms are cohered. During the first stage at the G2, you can see uncondensed sister chromatids arms are cohered, and after when they are cohered further in the prophase, the cohesion more cohesion molecules are attached, and it makes more condensed structure. During the middle of the prophase, condensed sister chromatids are free because the cohesins are released from the chromosomes. Uh, Cohesion remains only on the centromere. Uh, why only on the centromere? So the centromere can attach both the sister chromatids. During the anaphase, when the cohesins are released from the centromere, both the DNA chromatids are separated from each other. I hope this is enough for today, and I hope it will be easy for all of you. Thank you very much for today. Allah Hafiz.